the purpose of this lab was to isolate the GAPC2 gene in different plant species. In order to do this, we performed DNA extraction, PCR, gel electrophoresis, quantum prep clean spin, ligation, transformation, mini prep, and then sequenced and analyzed our DNA. We started by preparing the gDNA on basil and strawberry. With a razor blade, we cut into small pieces 60 milligrams of strawberry and 70 milligrams of basil. Once those were ground, we added lysase buffer to a microcentrifuge tube and ground that with a micropestle. More lysase buffer was added to the mixture, then centrifuged for 5 minutes. Due to centripetal force, there was a pellet that formed at the bottom of the tube, leaving us with a superdent on top. That superdent contained the DNA, so it was pipetted off and then mixed with ethanol lysase buffer and added to a column collection tube that was again centrifuged. This step was done to ensure the DNA would bind to the column. The collection tube was then placed into a column and centrifuged for another 1 minute at full speed. The flow through from this process was discarded. 3 washes were performed with a wash buffer to ensure that all the ethanol was dried off the DNA, and then centrifuged again. Water was then added onto the membrane of the DNA. This time the flow through contained our gDNA.
So after that, we did a first round of PCR, and that was required to test our Arabidopsis gDNA, P-gap plasmid in water along with our 2 plants. The gDNA acted as a positive control to make sure throughout our experiment we did not disrupt the plant DNA. P-gap was also used as a positive control to make sure we did not destroy the plasmid throughout our experiment. Water was used to ensure the contamination, no contamination occurred. To prepare our first round initial PCR, we made a master mix consisting of polymerase, DNTPs, buffer, and salt. The blue primer was added later to the mix, specific for the GAPDH gene. The first round PCR primers targeted for a section outside of the target DNA sequence. This master mix, the primer, and the 5 PCR samples were mixed in their respectable tubes and placed in a thermal cycler to run for a few hours.
Following that was our initial round of electrophoresis. To do this, we created a gel mixture that solidified and cooled in a gel tray. Then we were able to run our PCR products, which the PCR products were all mixed with 5 microliters of dye, which bound to the DNA so that it could be seen under UV light later in the experiment. The products were then pipetted into their respectable wells toward the negative side of the gel phoresis and then ran toward the positive side. The results of that, our negative control water did not generate any bands, meaning that throughout our experiment there was no contamination. The P-GAP and the gDNA did generate bands which we were expecting, and that told us that throughout our experiment we did not destroy the DNA. And then our experimental DNA did not generate any bands. In order to get rid of the unincorporated primers, we had to use or perform exonuclease. Exonuclease was added to only the 2 plant samples and incubated initially at 39 degrees Celsius and then again at 80 degrees Celsius to inactivate the exonuclease.
And you can see here, this is a picture of our initial electrophoresis, and you can see in these 2 spots we should see bands where we do not. So after that, we did a nested PCR. This is similar to the original PCR we did in the first round, except the primers in this round annealed to just outside the targeted DNA sequence rather than the entire DNA sequence as in the last PCR. A master mix was created and combined with our PCR products and the nested primers, then incubated for several cycles. This process allowed for denaturation, annealing, and extension to occur.
Following our nested PCR, we ran another round of gel electrophoresis. To create our gel, we again mixed the Argos with TVF buffer, let that cool in a gel tray, and then inserted our PCR products. We let those run until they were about halfway across the plate. Based on the results of this second gel electrophoresis, we decided to continue with strawberry. The strawberry plant or DNA needed to be purified with leftover dNTPs, polymerase, and primers. And to do this, we performed quantum prep PCR clean spin.
So after that, we did some blunting and ligation. For this experiment, we decided to blunt the ends of our DNA rather than leave a sticky end or overhang. Blunting it creates a free 3' end for the PCR products to bind to rather than the overhang sticky ends. Once the ends had been blunted, the PCR had to be ligated into a cloning vector, PJET 1.2. To do this, a small amount of PJET 1.2 and ligase was added to the PCR products and kept on ice to ensure transformation worked efficiently. This process joined two pieces of linear DNA into a single stranded piece.
We chose PJET 1.2 for 3 main reasons. It self-replicates, it has a high copy number, and then it has a selectable marker and control mechanism. It's also resistant to ampicillin, so it would allow us to know if our PCR was inserted correctly. Following that was transformation. This is the process where we inserted our vector with our PCR product into bacteria. For this lab, we chose E. coli. In order to do this, the bacteria cells needed to be opened to insert our vector. This was done through heat shock. The cooler transformation products were streaked using the streak plate method onto a warm agar plate. They were incubated overnight after that. The fast contrast between the cold transformation products and the hot plate allowed for the cell walls to open and our vector to be inserted.
So after that, we had to isolate our colonies. So after they grew over a week's span, there was a mini prep performed. To do this, isolated cell colonies were selected by the instructor and grown in a medium broth. You can see here our selected cell colonies that we had to choose from. The plasma-containing bacterial culture was resuspended with solution and centrifuge. The next step was to perform restriction digest, in which the enzyme BGL2 was used to cut out the inserted DNA within the plasmid. This process required us to make another master mix similar to the last, but this time with the enzyme and a buffer.
After that, our final round of gel electrophoresis was performed. Based on the results, we chose colony 4 with the best results. This colony went through DNA sequencing after being sent to microgen.
You can see here, this was our electrogelophoresis of our final DNA product, and it's very hard to see, but there's a light bar right here indicating to us that DNA was present in the sample. So then finally, after our DNA sequence was returned to us, we analyzed the results. Most of our hits were between 80 and 200 base pair lengths, as you can see up here. The purple indicates 80 to 200 base pair lengths, with some others being between 50 and 80 base pairs. Our E value was 1 × 10⁻⁴. Our query coverage was 94% and our identity percent was 97. Our sequence did not align with our expected GAPC gene and instead best matched a synthetic construct that was used generally for swine flu detection. That is kind of the profile of the sequence we ended up getting.
In conclusion, we attempted to isolate GAPC2 gene in both basil and strawberry plants by performing DNA extraction, PCR, gel electrophoresis, quantum prep clean spin, ligation, transformation, mini prep, and then sequencing and analysis of the DNA. Although this was not achieved, the 8-week-long process taught us laboratory techniques and protocol that we can carry over into future research on various gene sequences.
Thank you. Any questions?